Okay, so I think we're all set. So we will start today by reviewing a lab on Lima, and that's where we'll also end. Next weekend, which is Sunday the 4th, I believe, on December, that's when we will review. Right, so that's when we'll review the solution to homework 6, which is the gene set enrichment analysis for the pathway. So we'll review the solution, and I will try to see if we can review a topic of data mining on drug signatures, or we can talk about R packages, how to create them and build them. So one or the other would be an interesting topic. And the final solution that I will review is the homework 7, which is the Lima, which I already posted, and I believe that's due on December 10th. So on December 11th, that will be the last one that I will review. And I think on Saturday, and let me just make sure I get the dates right, so I don't have a calendar in front of me. So I'm not sure why it's taking so long. Anyway, so the next Sunday we'll review the gene set enrichment analysis homework, most possibly drug clustering and drug signature. And the last session I will review the Lima. And I think one of the team is presenting on December 19th, which is a Sunday. And other team is December 11th. I think it's your team, Angela, but I haven't got the exact timing. So if you're able to send me the time with the dim invite, that will be great. And I also haven't heard from the other third team on when they're planning to present, but I will double check my emails. But I will also not be available on December 12th from 2 p.m. onwards. So I know we have one presentation on 11th, one on 19th, and the other one either I don't know yet, or you guys haven't sent me the email that I haven't checked. Okay, so that's what's outstanding. And today we will review the time series lab data from Lima user guide, and what we will review is from chapter 7, I believe, and this is where the section 11.4 we will work with the estrogen data. There was an additional lab that was also loaded for the time series with Lima for the Drosophila data. But I knew that using Windows, you guys will run into some trouble uploading the package, or even though you're able to upload the package, the objects are no longer valid with the version of R that we have. But I will go through that topic briefly. But let's just review first the topic of Lima and one of the labs using the estrogen data, and that's in your Lima user guide. So once you are able to go through that example, it will help you on your homework, and then we will try to see what went wrong with the data set with the time series lab that I also have uploaded in doc sharing. So we went through the design matrix and contrast matrix. I think we were focusing on the 2 color arrays, and by the time I started ending the lecture, I think the dim session timed out, and maybe some of you haven't seen the last part where I was talking about how to set up a design matrix for an Affymetrix array. So here's an example, just so we can catch from there and try to relate to the lab that we will do. So they had 4 samples, okay, and under each of the samples they had different replicates. So sample A had 3 replicates and sample B had 4 replicates, and these are, it doesn't say that whether these replicates are done at different time points or not, but we're assuming it's a single time point for now. and for a single time point and 4 different samples, we want to see that how a design matrix can be set up, which describes how the experiment has been done. And contrast matrix is what do I want to compare with what. So I may want to compare sample A with B or sample D with B. So that will be the contrast matrix. And as far as design matrix is concerned, the Affymetrix single color array is much easier than your dual color array, because in dual color array in the last lecture you saw how we have to use 1, -1 depending on the die swap. But in Affymetrix array, basically each time an array is present in the grid, you denote it by 1. When it's absent, you denote it by 0, and we'll show you in the next slide. And the other part that's important in your design, along with your design matrix, is to understand the number of parameters. So the number of parameters in Affymetrix array will be the distinct sample types. In dual color array, it's distinct sample type -1 because of you only want to use independent parameters. But in Affymetrix array, it's simply the distinct sample type. So I have here 4 different samples, A, B, C, D. So the number of parameters will be 4 for me. So I can compare A with B, or B with C, or C with D, or D with A. So these are the number of parameters, A, B, C, D. So that's your parameter. So let's just go to the next slide that shows you how your design matrix is set up. Sorry. So on the design matrix, so these are your 4 arrays, A, B, C, and D. So when you look at A, so Y1, Y2, Y3, you'll see the first 3 rows here will be 1. So each of these columns here are representing your sample type or your parameters. So as I mentioned, there are 4 different sample types, so you have 4 parameters. So parameters go into the column, and your samples or your arrays go into the row. So if you had 10 distinct arrays, so 1 to 10, so you will have 10 rows, and the 4 columns that you have will represent the 4 parameters. In your scenario, the parameters are the distinct sample types, A, B, C, and D. So when you are by design matrix, you are showing the model how your arrays have been designed. So it's pretty intuitive. You can see it from here. The first 3 rows only belongs to parameter A. So 1, 2, 3. So it's 1 for in relation to A. So if your second column is B, third column is C, and fourth column is D, it has nothing to do with your parameters B, C, and D. So it will be 0. So only the first 3 rows here ends up here. In the second one, you will also have, because this is column B, you will have 3 1s. On the third one, you should have 2 1s because there are 2 arrays, because this was column C. And finally, in the column D, at the very last 2 rows, you should have 2 1s. So anytime you have a 1 that represents a presence of an array under the column, which is your parameters or your distinct sample types. So that's how your design matrix has been set up, and you multiply that by your parameters. So the model outputs the different values, which is basically it's estimating the Y1 to Y10 values for all of your 10 arrays, and you do that by setting up first a design matrix. So design matrix is easy for Affymetrix array. And the other concept is the contrast matrix. So contrast matrix is basically what you are comparing, what 2 arrays you are comparing, or you can compare 2 different time points. In our lab example, we'll show you that. So if you're comparing A with B, so if you have your 4 columns here, so that will be A is 1, and you're comparing B as a minus, so it will be 1 minus 1. It has nothing to do with C and D, so your third and fourth column will be actually 0. In the second row, if you're comparing A with C, so your A will be plus 1, B is 0, it's A minus C, so C will be minus 1 and D will be 0. And then what you actually do, so that's similarly how you did the design matrix, where the columns are the parameters and the rows are the arrays, the 10 arrays. But in order for this is how you start building your contrast matrix, but you will transpose it. So it will become 1 1 -1, 0, 0, -1, 0, 0. So in Lima, the contrast matrix is the transpose of the above. This one helps you to visualize what two things you're comparing. So if you're comparing A with B based on your design matrix, it will be 1, -1. C and D has nothing, no relation, so it will remain in 0. I already explained A minus C part here. Then you simply transpose that becomes your contrast matrix. So A minus D similarly would be 1, 0, 0, -1. Right? 1, 0, 0, -1, A minus D. B minus C would be 0, 1, -1, 0. So you can figure this out from this example. Then you transpose that, and that becomes your contrast matrix. So much on the design matrix and contrast matrix. So now let's get into the code. So this will be using the estrogen data. Okay. So I exchanged some emails with Angela, and I think she was the only one I heard who were trying to look up the time course lab that I posted on, how we installed the packages and how it didn't, work, and how it was built on a different version of our object. But I will come to that later after I end up explaining, how we work with an estrogen data from Lima. So let's start with the estrogen data from Lima. So again, that's from your Lima user guide, which is uploaded already in your Blackboard, and that's exactly, I'm replicating section 11.4 to help you do your final homework 7. So there it's a different data set, of course, and I ask you for different comparisons. But let's try to understand how we do the design and contrast matrix, and how do we do the Venn diagram between what is up and what is low, and there is a concept of F statistics and different things that are a little different, which we haven't covered before. So for the estrogen data, the different packages that we need is we need the AFI package to construct the expression set object. Estrogen library, it's already a package that's in the bioconductor, which they have the data sets available. We actually don't have a similar package for in bioconductor for the Drosophila data set. That's why we ran into problem. But the estrogen data is actually available in bioconductor as a package, so you can simply source it and do a BioC Lite, right, to import this package, and that's what I did. And 8GU95AV2, so this is the CDF file you need because the estrogen data was performed on this platform, on a human 95AV2 version platform. So that's the CDF file that you need to import. And Lima is the package that we'll be using to build our linear model and base fit and our DE analysis. So let's import the Lima library, then import the AFI library to construct your expression set object. I also import an annotation for the 95AV2 platform, but I don't think so I use the probe set to annotation. But that's something we have done in the previous homework. So should we need to, you can do that also. So this is the library for the annotation, for the CDF file. And finally, this is the data set that we'll be working with. In the past, we have worked with ALL data set, which was also in the bioconductor as a package. The estrogen data set is also available as a package. The Drosophila is not actually available as a package, and we ran into a bunch of problem working with that. But the original data sort is available, so you can still circumvent that and work with it. But that's the topic I will cover at the end of the lecture. So library estrogen is what we are working with. So let's import the estrogen package. So I did not have it at the beginning. So for your case, you actually have to use the source command and import the estrogen. And I also did not have the 95AV2 CDF file. So both of those, if you don't have, make sure you source and use the BioC Lite to import the estrogen package and the AV2 CDF package. So now let's look at the estrogen data briefly to understand what the data has been designed as and what do we need to analyze. Okay. Yeah, I can see the chat box. The 11th hour presented to. Okay, so Joseph and Angela, both of you send me a dim invite or any other invite that you have, you're using to share your computer and your PowerPoint slides, because I think I don't have the exact times in my calendar book. So December 11th will work for me. I believe it's a Saturday, and December 12th is a Sunday, right? And December 19th is the last group. So both of you, please send me an invite, an exact time frame on December 11th, an EST time that works for both of your group. So maybe hopefully if you can do it back to back, one after another, so that way we don't have to book two individual time frames. So if you would kindly send it, that would be helpful. If you email me the project, the PowerPoint slides ahead of time, I will upload it in Blackboard under project, because I don't know whether you guys all have the privilege. So maybe on December 10th or 11th, just a day earlier, you can. Oh, Joe and you are in the same group. So I think Bill's group is the one I think I haven't heard. So you guys are probably J. I don't know the abbreviations. I know Susan's group has it on 19th. Your group is on the 11th. So there's another group I think led by Bill and his and other members there, whom I don't think I know the time. Okay, thanks. So that works, Ed. So we're good there on 11th and 19th. So the other group doesn't have a date and time yet, but let me verify with them. Okay. So the estrogen data, according to the VigNet, we know that when a package gets created, they created different folders and etc., etc., and then actually it's stored in a folder called ext data. And when we go through a simple exercise of what we went through in creating the package, it's a different thing that will help you. It's a more of inner workings of R than bioconductor, but it's an interesting topic that you need to work through as you continue your assignments with R going forward. So it's available in the exdata tdata subfolder. All the data is there, right? So let's look at the directory. So what it's basically saying, after you imported the estrogen package in my Windows and C: program files, chart T, document setting, etc., wherever my R is, so there's a standard library folder. So anytime that you're actually importing a bioconductor package directly by using the BioC Lite, this is the default path under the library. Then it creates additional subfolders. So estrogen is the package I created, and it created ext data folder, and under that folder it actually stored all the files. So if you're not using bioconductor, but someone has created a package and they may be keeping in their website, so you have to be careful when you are actually installing those packages into your Unix and into your Windows environment. I find such things, packages, copy and usage is much easier if you have a Linux or Unix, but it gets complicated on Windows between the source files and the binary files. It's a little complicated topic which I cannot cover in one lecture, but I'll try to briefly overview that today. So as I use the BioC Lite, this is the default directory. Under library, it created the estrogen folder for the package, and ext data is the subfolder under which the data is kept. Okay, so that's my data directory. It gives me the path where my data is kept after I imported the estrogen folder. So if I use a dir function on data directory, it tells me under that directory what are the different files. So under ext data, what are the different files that this package has imported for me. So here I see all the cell files. So I see 1, 2, 3, 4, I think about 10 cell files, right? 1, 2, 3. 4, 5, 6, 7, 8, I think. Sorry. So I have 8 cell files, and it also has a description of phenodata.txt, which probably tells me which cell file is for which condition. Okay. So now what I want to do is I want to read this txt file, which is a simple, the way we read any of our phenodata in the past. So it's let's read that phenodata text file. And let's see how it looks like. So this is how it looks. You have so each time you cell file, you see low 10, 1 and 2, that means it's a replicate, right? So it's similar to what we showed you in our design for the one color array. Okay, I think I don't have to go there. So you have a replicate of the same condition. So you have a low 10 cell file, same sample but replicated twice. And over here, so let's assume that you did not treat this sample with an estrogen. So it was the estrogen was absent there at 10 hour. Then you had another sample with 2 replicates where you actually treated that with estrogen. So estrogen is present. So this is your reference baseline time point at 10 hour. Okay. Then you took so you treated your tissues with the estrogen, and at 48 hours you withdraw the second set of samples, and you have a replicate here. So that's for 48 hours, where estrogen you did not treat with estrogen, and the other one is, let's say, you treated with estrogen. So basically your condition as far as estrogen is concerned is either present or absent. So that's your 2 types of samples, and you have 8 cell files. And out of those 8 cell files, you have 4 distinct ones, because for each of this condition you have a replicate. So that's how your pheno data looks like. So from here we have to get into a design matrix, and we have to go into a contrast matrix, and there is some notation in the lab they set up. So let's just try to look from the perspective of what is that they tried to compare. They probably tried to compare the cell files that are present at 10 hours against the cell files that's estrogen present at 48 hours. You could have compared absent with absent. You could have compared the absent at 10 hours with the present at 48 hours. So there are many different comparison and combinations that's possible. In the lab, we will focus on comparing the files where estrogen was present at 10 hours against estrogen that's present at 48 hours. So there is a dimension of estrogen itself between 10 to 48 hours for differential expression, and there's also a dimension of time between 10 hours to 48 hours. So that's how your your pheno data looks like. So now we do our simple things, which is we use the read-affy to construct the expression set object based on the file names that we have in our targets. So that's our target. So for each of those file names, I will read them, and then I will construct an expression set object. Okay, so it's still constructing. So I think we're done. So we're done creating the expression set object AB. So now let's normalize, summarize, normalize, background, correct, and summarize by RMA. So that should be also very simple by now for all of you. So let's do that. So this is our pre-processing steps. So in traditional analysis we have done so far, from this point onwards we would have gone to do the non-specific filtering, then we would have done a simple t-test or multi-test or SAM, etc., etc. Then we would have mapped it to an annotation, or we could have done a hypergeometric approach, or we could have taken a GO approach, gene set enrichment approach for JESA. Here we're doing things differently by linear modeling. Okay, so up to this point is no different than what we have done till this date. So now you have your expression set object as corrected, normalized, and summarized. Then let's try to understand the notations, which gets a little tricky. So you have 8 arrays, right? So 8 cell files, 10-1, 10-2, 48-1, 48-2, and then 10-1, 10-2, and low 48, low 48-2. So you have highs and lows. So highs means maybe estrogen is present, and low means estrogen is absent. And 10 means the things that were done at 10 hour, and 48 means things that were done in 48 hour. Okay, so this is your matrix. So you have your 8 cell files. These are 4 of them are done at 10 hours, 4 of them are done at 48 hours, and within each hour you have replicates, 2 replicates, and within each replicate it could either be a condition of absent or present. So pretty descriptive. You can visualize what's going on in your experiment just looking at your pheno data. Okay, so now that we have done that, so let's establish some notation, and I took some notes. So in the notation what we are saying, anything, the estrogen when it's present or absent at 10 hours, right? So 4 samples are at 10 hours and 4 samples are at 48 hours. So when I say E10 or either small cap E10 and upper cap E10. So that's the samples that were withdrawn at 10 hours. When I say lower cap E48 and upper cap E48, these are the 4 samples that were drawn at 48 hours. So 4 and 4. So you have 8 of them. Then we know that each of these, so when an estrogen was done at 10 hours and that was absent, so lower cap E is equal to absent and upper cap E is equal to present. And I follow the exact notation that's in the lab. So it's easier for me to explain and easy for you to later go back and understand what happened. So when E, which is lower cap E, which is absent, done at 10 hours, so there was 2 replicates of them, right? So there's 2 of them here, what I'm highlighting, 2 of them there, what I'm highlighting, which is upper cap E10. So 2 and 2 is 4. So you basically have 2 plus 2 here, and then you also have 2 plus 2 here. So that describes your 8 files, and that's your notation. E is absent and R is present. Then if you want to find out the category, so now this is a good way to represent a categorical data as an object, which we will use factor. So factor in R represents your categorical data, and it also sets up the level under each factor. So the level tells you what's distinct under your category. Okay, so if you have something called male, female, female, male, male, female, so these are different categories. The levels are only male, female, so that's your distinct. So that's the same thing I'm doing here by notation of 1, 1. What I'm saying, there are 2 1s. So this is your 1 and 1, right? And this one is your 2 and 2. So basically I have 2. By saying 2 and 2, I am indicating that I have a replicate, which I'm labeling as 2 and 2 for the one that was uppercase E10. By 1 and 1, I am saying that this is the lowercase E10, which is the estrogen absent data. So basically you have 8 samples, right? So when you have write from 1 to 4 and you duplicate each time, so you have a label, unique label for each of your 8 samples. So 1 and 1 represents your E10, 2 and 2 represents your uppercase E10, 3 and 3 represents your 48 hours absent, and 4 and 4 represents your 48 hours present. So E equals to present. So that's your factor. Okay, and then on whenever your E and E is same, so that's what does label gives you? Label gives you distinct under your factor. So 1 and 1 should be a label of 1, 2 and 2 should be a label of 2. So basically your 1 and 1 belongs to the same condition of E10. So first you set up the factor, then you set up the label, which is basically getting the distinct out of any 2 that are similar, and then that gives you a very good description of your 8 cell files and the grouping of them. So now let's look at treatments, how it looks. So your treatments, so these are your 1, 2, 3, 4, 5, 6, 7, 8, right? 8 different cell files, and your levels are telling you what is the grouping or the distinct about it. So E10, 10 hours, estrogen absent, estrogen present, estrogen absent at 48 hours, 2 of them, estrogen present at 48 hours, 2 of them. Now you have your 8 samples. So you are just labeling your samples, so then you can use it into your design matrix. So that's labeling. Now if you look at the slides that I showed you here, and let's just go back to the slide, which will helpful. When you looked at the slides by Natalie Thorne, you can, so this is for 2 color arrays, so we're not interested on that. So this is the single color array, right? So they're very similar. You have A, B, C, D. In our case, we have A and B, which is estrogen at present or estrogen absent. So that would be our 2 different sample types, and then we will actually have 8 different arrays going that way. So for the 10 hours time point, we'll have 2 replicates. For the 10 hours time point, for 48 hours time point, we'll have 2 replicates. For if you have absent here, so sorry, if you have A as estrogen present, right, at 10 hour, then we'll have 2 replicates. B could be estrogen absent at 10 hours, so there'll be 2 replicates. C could be estrogen present at 48 hours, 2 replicates, and D could be estrogen absent at 48 hours and 2 replicates. So by doing these 4 arrays of this, 2 samples each, you can also represent your design for your single color array. Just forget the 3 and 6. Then when she showed you the design matrix, each time that the A was present, we had 1s here. Each time B was present, we have 1s there. So the parameters here were actually number of samples. So I had 4 parameters, A, B, C, D. So that's how it was done in the background that I gave you between contrast and design matrix. So if I were to follow her notation in our scenario, we can say that since we have 8 arrays, right, and we have 2 different sample types, so our parameters will be 2. So the matrix that I'm thinking we will construct will be an 8 by 2 matrix. So 8 of the rows will tell you 8 cell files, and 2 of the columns will tell you either the sample types. When you look at it, forget the 10 hour and 48 hour, either in those sample you had estrogen that was present or estrogen that was absent. So your parameter could be 2. So your design matrix can be represented in an 8 by 2 matrix. Okay. And another thing that when you saw when this modeling was done, it was usually an equation of y is equal to bx, right? Something like that. So y is what you're trying to estimate for your 8 arrays, b is your design matrix, and x was your parameter. But normally in linear modeling, you usually try to figure out a formula called ex bx plus c. In many cases, this c is actually called a bias or an intercept. Okay? So to factor in this bias, which is the c, you actually do an additional factor, which is your bias of 0. So I can label it as 0, 0. Your 1, 1, 2, 2, 3, 3, 4, 4 that you have in the factor is the same thing that I initially explained, where you're labeling your 8 samples. So here's the label for your 8 samples, and one is duplicating here because it's a replicate, and I'm adding the 0, 0 to take into account for the bias. If you do not want to do your linear modeling by using the bias or intercept, you don't have to do that. But in most of the example I have seen, this is the modeling that you're actually trying to set up. So that's the best practice. So includes the bias. So I also do that. So I label them as bias, and E10, according to the notation I just explained to you, will represent 1, 1, and uppercase E10 will be 2, 2, and hence so on and so on. So you can do that. So that's your treatment prelim. So let's look at that. So as far as factor is concerned, it looks like it will be 10 of them now. 1, 2, 3, 4, 5, 6, 7, 8. So 8 of them are samples. 2 of them are for bias. So bias itself was replicating. And these are the different levels that you have. So now that you have your factor, you can, based from the factor, you can actually design your matrix by using this notation model.matrix, where you actually take the transpose of the treatment prelim, and that automatically designs a matrix for you. You don't have to be fancy in using the model.matrix. You can actually use a very simple column bind and row bind function that we have showed you at the very beginning of the class. And let me just explain to you what I mean by that. So when you draw out your design matrix, and if you know that end of the day this is the matrix that you have to design, where all the biases are 1, right? So then your first column, which is just the 10-hour absent for estrogen, so you know your first 2 ones will populate here. The second column will be 10-hour estrogen present, so your second 2 rows will be 1. Third one is 48-hour estrogen present, so your third row will be 1. And finally will be your 48-hour estrogen absent, or present, because it's uppercase E. So your last 2 rows will be 1. So if you know that's the matrix that you're trying to construct, you don't have to use a fancy notation of model dot matrix. You can just use a column bind and row bind to get to your matrix. But that's how, if we would have used the slides of Natalie Thorne and the way we understood our factors and labels, that's how it would have worked out. But let's just see how the lab actually went. The lab gives you a direction to get to the design matrix from the contrast matrix. So in the normal flow, what we have learned, that you first establish your design matrix and then you get to your contrast matrix. When we propose the homework solution, I actually have developed the solution that way. So first I try to figure out, okay, what's my design matrix? What are the arrays? What are the parameters that I have? And where do my ones go? So that's a simple thing. That's your design matrix. Then I try to see, what am I trying to compare? Am I trying to compare the 24-hour with 6-hour, 6-hour with 1 hour? And that's when I do the contrast matrix. But in this lab, I think we try to first see what is the contrast matrix, what am I trying to compare, and then reverse engineer to build the design matrix. So if you look into this particular example, what they are interested to compare is the 10-hour sample, right, with the, I'm sorry, okay, compare estrogen, which is only present between 10-hour and 48 hours. That is, we are interested to compare uppercase E10 and uppercase E48, which is for present, right, only. Not interested in lowercase e10 and lowercase e48. Okay, so that's the first thing you have to see here. They're not interested in these two comparison, but they're only interested in e10 and e48. So if you are only interested to compare between e10 and e48, so for the contrast, what will happen, you have 8 samples. So out of your 8 samples, you know 4 of them belongs to E10 and E48, and other 4 of them belongs to lowercase e10 and lowercase e48. So instead of doing a 8x2 matrix, if you're limiting your search to uppercase E10 and uppercase E48, you'll be only needing to do a 4x2 matrix. And that's exactly what I show here, the 4x2 matrix. So you have a 10-hour absent, 48-hour absent, and you have a 10-hour present and 48-hour present. So if you're interested to compare only E10 and E48, none of these will apply because these are still lowercase e10 and e48. So I'm just not showing the replicate here. You will actually get a 8x2 matrix when you include every single array, and that's your design matrix. In contrast matrix, it's a subset where you're actually grouping the distinct sample types, your parameters. So instead of a 8x2 matrix, here you're concerned with a 4x2 matrix. And in this 4x2 matrix, the lowercase e10 and e48 doesn't apply when you're comparing e10 with e48. So these will be zeros. But your e10 applies with e10, so that will be 1. And your e48 applies with e48, so that will be 1. So should you think of a contrast matrix that goes between E10 and E48, it will be a 4x2 like that. And should you think of a contrast matrix, which this lab doesn't do, between lowercase e and lowercase 10 and lowercase e48, it will also be a 4x2, but instead of, it will be 1 and 1 here and 1 and 1 there, right, because this matches, and lowercase doesn't apply here, so all of them will be 0. So that's a conceptual visualization of your contrast matrix when you're comparing between these two parameters. Now, another thing that we are also concerned here, that not only I want to compare between E10 and E48, I also want to see what's the effect from 10 hours to 48 hours. So E10 is a parameter or a coefficient. E48 is a parameter or a coefficient. Your time factor, which is going from 10 hours to 48 hours, that's also a coefficient. So what I can actually do, I can slightly alter it to, let's do it by this way. right? And I can actually write it like that, and then I can do another grouping of time. So let's say I will say when time is 10 hours, I want to notation is 0, and when time is 48 hours, notation is 1. So if time is another coefficient, so I've grouped the 10 hours here, so it will be 0 here, it will be 0 here, it will be 1 here, and it will be 1 there. Then for every single linear model that you have an intercept called bias, which is always applicable. So these are your 4 coefficients that you're dealing with in the example they're giving. You're dealing with E10 with E48. So that alone, see, you don't know, because sometimes you can have a E10 and E48 given on a single time point. So this is how you will do your compare matrix, E10 and E48. And then, if you happen to be collecting, so let's say let's say you're looking at an experiment where it's nothing to do with 10 hour and 48 hour. You have 4 different replicates. So then if you can simply compare with E10 and E48, so that will become your 2 parameters or 2 coefficients, and you will indicate 0 or 1 according to where it's present or absent. But you have a different dimension on your experiment where you not only have to worry about the estrogen present and absent between 2 samples, but what's the effect between 10 hours and 48 hours. So this is an example where you actually did not see hair. Here it doesn't tell you, I'm sorry again, the model that you are showing here doesn't tell you that whether the samples, this is done at 1 hour and whether this is the 6th one is done at 4 hour. It doesn't tell you that. It just tells you I have 4 different samples. It's very well possible that each of those samples were replicated within a single time point. So you don't have the dimension of time from the example in how you set up the design and contrast matrix. But over here in the contrast portion, you actually do have a dimension of time. So 10 hours are grouped as 0, 48 hours are grouped as 1. You have a bias component, which you're always saying as 1, because you're trying to solve the equation of nature y equals to bx plus c. C is the bias. So that's your 4th parameter, and first 2 parameters is your 2 sample times that you're trying to compare. Okay, so then when you're trying to contrast and make a contrast matrix out of your treatment, you'll include all of your 3 dimensions, right? So you'll include the time dimension, 0s and 1s. So as I said, the first 2s are 0s and the last 2s are 1. So this represents your 10 hour and this represents your 48 hours. So that's your time dimension. The uppercase E10, which is your sample for estrogen present, so it's only present on the 2nd row. The 3rd and 4th row it's not present. So you have 0 1 0 0, so that's very simple. E48 should be 0 0 0 1. So this column bind. So what does C, if you do a column bind, so 0 0 0 1 represents your 2nd row, 0 1 0 0 represents your 1st row, and 0 0 1 1 represents your 3rd row, and bias will be defaulted to 1. And that's how you do your contrast matrix. So you use the function contrast on treatments by doing a column bind, and column bind is nothing but combining each of those particular columns. So bias by default gets assignment as 1 1 1 1. So that was your 2nd column. The time column was your 2nd column, which is 0 0 1 1. I just drew it differently. Your 3rd column was E10, which is 0 1 0 0, and your 4th column was E48, which is 0 0 0 1. So that's the most complex part. Even I find it very challenging. I mean, you might find it easy. In constructing my design and contrast matrix, you really have to know a lot about your experiment, how it's set up, what you're comparing, and you just cannot arbitrarily set up these 0s and 1s, because that will end up resulting in nothing. So this is the most challenging part and requires a really rigorous and thorough understanding. So let's look at the, and I think I'm going to reduce the things because it's barely visible this way. You cannot see the whole picture. Okay. So that's your contrast matrix. Sorry, that's not that. That was mine. Okay, so I think I did not cut and paste something correctly. Let me just see. contrast treatments. Okay, so now I did that. So now let's look at the contrast treatments. So now here you have your time portion, which is your first column. So this is your 10-hour grouping and 48-hour grouping, and 10 estrogen present and estrogen absent. Why it's 1 here and why it's 1 there, I already explained you here, right, on this particular section. So that's the contrast. So you have 3 parameters. You have 10-hour estrogen present, you have 48-hour estrogen present, and you have time, whether it's a 10-hour factor or a 48-hour factor, and then you have a bias, which is 1, 1, 1, 1, which is always there. So your treatments, if you want to check it again, which you created earlier, now becomes, you have your contrast matrix plus your factors and your levels. So when you do the, so first you started with treatments where you only set up your factors, which is 8 sample types, then your levels, which is your 4 different unique sample types, then your contrast is what you're trying to compare. You're only interested to compare E10 with E48 because it's a time course data. You have a third parameter, which is the time, and you group the time. For 10 hours you have a notation of 0, for 48 hours you have a notation of 1. Okay, I think we made some progress, I hope. So that's your contrast. So what will be your design matrix? So design matrix will be, you have 8 samples, right? So it will have 8 rows, and it will be an 8 by 4 matrix because you have 4 parameters. First is the time parameter, second one is your estrogen present at 10 hours parameter, and estrogen present at 48 hours parameter. So these are your 3 parameters plus the bias, which is always present in each of your samples. So you will have a 8 by 4 matrix. 8 samples in rows and 4 parameters to compare. So let's just check it out, that if that's what we exactly get. So our treatments already contains the contrast that we want to build, and what are the different levels, sample types, and what are the different samples I have. So I will get 8, right? I will get the 8 rows from here, and the 4, which is the column, I will get from these 3 of these parameters plus bias. Then you can use this model.matrix. If you don't want to use that, you can simply construct an 8 by 4 matrix that you can draw out on a piece of paper and do a column bind if that's easier for you. Okay, so now this is how your design matrix look, right? See, that's the intercept, right? So you have 8 samples. So on the first column, all of them will be 1s. So that's the intercept. Then your time, so if it's 10 hours, then it will be 0s, and it will be 48 hours, it will be 1s. So that's how your 0s and 1s for the time factor. Then E10, so 2 of them are present on the first, then 2 of them is present on the second. So that's your 4 different samples for the E10 and E48. The other 4 is your small E10 and your small E48, which you are not comparing, therefore it's not on your design matrix. So here you actually went from your contrast matrix to see, what is that you're interested to compare, and then came up with an accordingly relevant design matrix, because you're totally not interested in your small E10 and small E48, which you're not comparing. You're only comparing your estrogen present between 10 and 48 hours, and your third parameter is your time between 10 hours to 48 hours, and then you have a bias. So that's your design matrix, which is a 8 by 4. So we talked about that. So 8 samples in rows and 4 parameters in the columns. So that's your design matrix. So now let's just label them instead of intercept and XYZ. So let's label this as intercept. Let's label this as time. Let's label this as estrogen present and estrogen present at 48 hours. Or E10, E48 will be sufficient. So over here I'm just labeling the column names for the matrix. So you can revisit that design. So you have intercept, time, E10 and E48. That's relevant. You don't have smaller E10 and smaller E48 because that's not what you're comparing. This is an example. You thought about your contrast matrix first, then you went into your design matrix. And design matrix is a 8 by 4, 8 samples for 8 cell files and 4 parameters that you're comparing. So then what we do, this is the second, so there's a concept of coefficient. So intercept is the bias coefficient. The second coefficient, which is the time, that tells you what's the effect that you have on your system for samples that went from 10 hours to 48 hours. The third coefficient measures what's going on with your estrogen present sample at 10 hours and what genes are up or down at your estrogen present at 48 hours. So these third and fourth coefficient will simply tell you between estrogen present at 10 and 48 hours what's upregulated, what's downregulated, which didn't have any differential expression, and the time effect will be figured out by the second coefficient, and intercept effect is present in all of them. So those are your four coefficients. Then you actually do this model fit by simply using the lmfit function, where you give your original data set, E set, which is what you got from your RMA expression. So you get your E set, so that's your original data set that you started. We don't do any non-specific filtering and IQRs and SDS and all that. So we simply take the E set and we tell that this data set or E set that I have, expression says object, is designed like the design matrix that I just built here. Ones and times and estrogen present 10 hours, estrogen present 48 hours. Sorry. So that's what you give on your that's where you actually are figuring out E is equal to Bx plus C. So C is your interface, your X is your parameters, B is your design matrix, or let's say D is your design matrix. So your base estimated model, base model, is equal to design matrix by parameters plus bias. So that's what it's going on. So all this is done by you, by the Lima package. So it did that very fast. Then now that you have your calculated values, based from those calculated values, now you have to use a contrast matrix to compare what you want to do between E10 and E48. So for that you design the contrast matrix, and contrast matrix should look exactly like this, but not the time factor, because I'm only comparing 10 with 48. I'm not comparing 1 hour samples, sorry, 10 hour samples with 48 hour samples. That's not my comparison. My comparison is E10 with E48. So my contrast matrix should look just like that. Simple. E10 with E48. So for the 4 sample types I have, I will have the first 2 as zeros, and because these are only small case E and small case so they will not be available, and the other one will be the present, which is uppercase E and uppercase 48, which will be available. Okay, so now what you do a combination of what you did for your design matrix and your contrast matrix, and you calculate a second model, second fit, which will be the fit 2, which combines your contrast and design matrix. And it's interesting that when you look at the sizes of how it wraps up, that your design matrix was 8 by 4, right, because of 4 parameters, and your contrast matrix, when you come to think of it, has to be a 4 by 2. Those of you know linear algebra, so this 4 by 4 will cancel out, so what we'll end up with is a 8 by 2. Right? So you you started with your design matrix 8 by 4, so the contrast matrix that you have will be 4 by 2. What we'll end up with is 8 by 2, and in 8 by 2 for 8 samples and this 2 is 2 comparison between E10 between E10 and E48. So that's how it works out. So that's your fit 2. So remember, the fit 1, when you'll do for your assignment, has to do with the design and your original data set. The fit 2 combines the contrast matrix, which is your comparing 10 hour with 48 hours. And that's what we do on the fit 2. And there's an enormous amount of theory behind Bayes and empirical estimates and posterior probability that it's well covered in a statistics class. So this estimates the probability and the p-values and t-values and etc. So let's look at the fit 2. So when you combine your contrast and your design, you get a model fit 2, and based on this model, now you're going to find out what's upregulated and what's downregulated. So this model, if you try to look at it, has different parameters that it calculates. The first one is, in your model, this was the design matrix. Your intercept was like this. You have 3 parameters that you wanted to compare because of in relation to time, but then you're also comparing E10 with E48. Then your contrast matrix was E10 with E48 plus your time effect additional. So that was your design. That was the design that went into fit 2. Then what it does, there is another thing it calculates as QR. So what QR does is it actually calculates the values of your intercepts for each sample. So this is the result of your y equals to, so let's say ax plus bx plus c. So this is the c, and this is the value of the bx, which is your design matrix multiplied by your parameters. So this is the different QR fit values that you got. But what you will be interested is to see that after your fit, what are the p-values of the different probe sets that you have. So when it's the so these are your probe sets, right, on the row wise, and when you did the estrogen present at 10 hours, that's the individual p-values, and when you did the estrogen present at 48 hours, that's your estimated p-values. And one additional point which I do not cover in theory, which is Bayesian statistics not only looks at the p-value, but it looks at the adjusted p-value by F statistics. So in the past, we have reviewed the adjusted p-value based on permutation t-test and multi-t-test, which is the multi-test package. But there is an additional concept where you can actually adjust the p-value based on F statistics. So you actually, for each of these p-value, you compute a log value. And I do not have the theory behind logs yet. And from the logs, you actually get into F adjusted p-value. So if your first p-value of the first probe set, which is 100 GAT, was 0.0175, then your adjusted F p-value would be 0.156. So try to understand the different parameters that fit2 actually outputs, right? So it outputs the original p-values. So from the p-value, it calculates the LOD values, and from the LOD values, for each of these probe sets, it calculates the F value, and then each of these probe sets, you get an adjusted FP value. And this adjusted FP value is what we will use our criteria to say what genes are upregulated or downregulated. And right, and then you start, it already tells you what is the design matrix that you started with, what is the contrast matrix that you started with, and these are the prior and post probabilities, which I don't also have the theory behind. Okay, so now that we have our fit, we have our combination of your design and contrast. So okay, now we have a model. So now how do we go to the next step? How do we go from the model to telling us what's up, what's down, and what had no change? So that's the final part. So you can use, this is what we use in multi-test, which is the Benjamin-Hochberg's model, and controlling an FDR. So they have a default FDR rate of 0.2% or something that they can, so you can do your decide tests on your model. You can use those p-values and adjust them based on the FDR, which we did on homework 4. But the new concept that they have in this, which is to use the F test of those p-values. So instead of using an FDR-based adjustment, you do an F test based adjustment of the p-values, and for that your method will be nested F. Okay. So we don't use the multi-test. You can, but in the assignment I would like you to use the nested if based adjustment of p-values. So you do that, and your results will be for each of those you have 12,000 rows, so you have estrogen present, you have estrogen present, so your nested p-value could be 0 for some cases or have some values, but what's the criteria? Do you want to get something less than 0.05, greater than 0.05? So first of all, the criteria they use here is the control probes that you have between your estrogen present at 10 hours and estrogen present at 48 hours should not have any differential expression. So let's find the p-value of those control probes, and my criteria will be I will have to use a p-value that's even less than that. So if your control probes I know are not having any differential expression, and your p-values for those control probes happens to be, let's say, 0.05, then I definitely want my criteria to be less than 0.05. So first of all, you get all the feature names that starts with AFFX that gives you the control probes. Then you summarize to say, okay, what's the mean and what's the median of my p-values for my control probes. So the minimum p-value is 0.000139. So in your criteria, you will use 0.0001. And then you will refit it, your result. So that's your criteria. And let's just show the summary of the results. So summary of the result tells me that by using this p-value criteria of 0.0001, which is adjusted by the f-test, between E10 and E48, I have 40 genes that's down-regulated, 76 genes that's up-regulated. Majority of the genes between F10 and F48 showed no expression. 116 genes were up-regulated just at E10, and 139 genes were up-regulated at E48. And that's where you just actually can draw the Venn diagram, and I ask you also what's up-regulated between 6 and 24, what's up only at 6 but not at 24. So you have to understand those relationships by doing a Venn diagram, which is sort of like a set of union, but the picture can tell you better. But let's just even understand, what does this 0 mean? So if I'm saying that I have no changes, so let's say you had a probe set, I don't know, 10180. Okay, so for that probe set at 10 hour, let's say the value was 7. At 48 hour, also the value remains 7. And when the estrogen is absent at 48 hours, at 10 hours is also 7, and 48 hours is also 7. So basically here, between 10 hours and 48 hours, nothing has changed. So there is no change. But how about both are upregulated? So if you had started at 10 hour at 7, that's your intensity value, and at 48 hours that estrogen actually became 10, right? So for the 10-hour sample, it's the estrogen is up, and in the 48 hours it's also up, the estrogen present. So both time points at 10 hours and 48 hours, your estrogen, actually I should not use E10 and E48. So this is present. So E I know is present, and so if 10 hour it was 7, and at 48 hour it's also 10. So that means in both are upregulated at 10 and 48 hours, right? Okay. So if you now draw, if you try to understand the summary result from the table I just showed you above, which is this one. So let's go back to done. And the way I would interpret that, I have to actually correct these notations for you guys. This is confusing. Okay. So what it's saying that if you do 87 plus, and let me just do the Venn diagram here. And where is the Venn diagram? Okay. So between 10 hours and 48 hours, if you add 87 and 52, that's 139. and you can actually see the 139 at E48, right? So 139 genes are upregulated at E48. So if you do 87 and 52 together, that's 187 and 52, 139. Okay, 29 and 87 is equal to 116. So 116 genes are upregulated at E10. 139 genes are upregulated at E48. Happens to be 87 genes that are upregulated between both at E10 and both at E48. So these genes were upregulated at your 10-hour sample. They also were up at E48 sample. But some of them were only up at 29 but not at 48. Some of them are only up at 52 but not at 10. So that's the diagram you have to interpret on your homework. What's alone at 48, what's alone at 10, and what's happening at both 10 and 48. And if you sum them up, your 87 and 52, that gives you everything that's up at 48. And if you sum 29 and 87, that gives you everything that's up at E10. So that's for your upregulation. This is what 0 means nothing happened, and -1 is for your downregulation. And for the downregulation, you can also draw a similar diagram, but instead of include equals to up, you say include equals to down. And I think in the homework, I tell you how to do it in upregulation. So if you add 29 and 11, so that's 40, right? So that lines up. And if you add 29 and 47, that's 76. And in additionally, it finds the Venn diagram, what's in common. Then finally, you can actually look at your top table to give you the output of each of your parameter, and you report it by 3 digits. So top table gives you what were the probes. So this is the t-test that you did for each of those probes. Then the p-value. Then this is the adjusted p-value based on the f statistics. Then there is also a b-value. But I highly recommend that you actually question the top table and study what are the different arguments and what is actually b and what is p-value, and how can you sort different things by b, how can you sort things by log fold change, and how do you report across coefficients. So I give you brief ideas and directions, and this is a complex topic, so it'll probably take you more effort. I believe personally this was the most difficult concept or homework to do, so continue your effort. And then when we review the solution on December 10th, we'll try to learn more from this. But on a basic level, try to go through this example as many times as you can. And then if you have further questions, you can ask me, because this is a complex topic, and there are many different ways to approach of doing the same design matrix or contrast matrix. But I will upload this particular case study that we just did. So start from that step, see what's clear to you, where it's not making sense, and let's just carry on with that. And the final part I wanted to discuss, which is this time series lab, which I additionally gave you. This also has very good example of how you're actually creating the design matrix. So if you go up from here, it tells you the design matrix, calculates the contrast matrix, and then you actually, instead of using the Venn diagram, you can actually ask between 1 hour, 3 hour, and other hours, what's up, what's down. So that's a slightly different way of doing it. But I ran into problem even running this lab, because when this is not available in Bioconductor. So the one way to do that is, if you look into this instruction here, what they're giving. So this is the source file for Windows. So this is a zip file that I first uploaded. Okay, so I did the zip file, so the package is installed. And then when you do a library, right, in R, by default your library is, and I showed you at the very beginning over here, which is C:\program for me and R2. version 10 that I have, and that's the library folder. So when you load something as a library and try to call it, it expects that the library package is available under this directory. But if in my case I actually kept my zip file that I uploaded from them at let me show you just very briefly. Okay, so web session 10. So this is where I kept my zip file, dosembryo1.on1.ozip under web session 10, and I'm saying the library should be under this libs folder over here. So when you create the package, and the library can be done under the libs folder, so that's what I did. My package is available here, the library should be here, and it's not available at CRAN, and don't use the BioC Lite for it. But then when I'm actually calling the library, make sure my library location path is exactly where I have put I have told my install package procedure to create the library at. Then when I select the data library, data draws embryo library, it's actually a pre 2.4 S4 object, because the version of R that I have is all built in S4 based object, but this particular library was built at an S3 object. So the recommendation is to create it from scratch, right? Or you can actually go to the website here where all the cell files are there, and then based on that cell file, just like you do for any homework, create your own data and create your own readafy expression set object, and then once you created that, then you can actually follow this tutorial from the design metrics and contrast metrics perspective. So that's pretty much what I have today to share. So let me upload it, and I believe the homework could be somewhat challenging or could be easy for some of you. So if you have questions, feel free to contact me. It's due on December 10th. And if you need additional theory, what's going on behind F statistics, why my contrast matrix is not making sense, ask me. I'll try to help you the best I can. And make sure you question the top table to understand what's actually going on and how the sorting is happening. But I'm just giving you as much guidance as I can. Take it as a template and try to build from there based on what I'm asking in homework. So hope it was somewhat helpful. So I'm going to end today's session. And okay, so those of you who have not one of the group hasn't told me about the project, right? I think we have one session on December 11th, which I need a dim invite from Syed or Angela. December 19th, we have the time. The other group, I don't know when they're presenting. So I'll look forward to wrap that up. I'm not available December 12th, though, at 2 p.m. So try to schedule it either at December 11th, right before or after Angela and Syed's group, or try to do it at December 19th, right after Susan's group. So thanks, everyone, and hope all of you had a good Thanksgiving break. And I have 2 more sessions left. And I will talk to you guys next Sunday. Take care.